Hey everybody, this video starts with scientists who were studying bacterial DNA, and I believe it could have been as early as the 1980s, possibly 1990s. The important point is it was relatively a long time ago. And what they found, these scientists who were studying bacterial DNA, is they found these strange regions of, bacterial chromosomes that, they were weird. The sequences were strange, and they couldn't figure out what was going on with these sequences. So what I'm diagramming here, these rectangles, I'm diagramming, imagine this is bacterial DNA, so double-stranded, just normal double-stranded DNA. And, if you saw the title of the video, you might have an idea of what I'm diagramming. These look like just checkered boxes, right? Alternating black and white. And, I could put a couple dozen of these in if I wanted to be accurate. But I'm going to end here. Okay. So, what they found was that some parts of the bacterial chromosome had these repeated sequences. And for now, let's just say the repeats are, let's say the colored boxes here are repeated sequences of about 20 base pairs in length. So the repeated sequences are identical. And they're found one after another in sort of a cluster in a certain location of a bacterial chromosome. And the intervening sequences, the spacer sequences, were also about 20 base pairs in length. Okay, I'll call these repeats. We'll call these spacers. And the spacers were unique. So, and let's say when they would find a region like this, there would be, let's say, about 20 repeats. That way you have an idea about how many repeats and how many spacers there are in one of these clusters. It's not going on for thousands of repeats, as far as I know. A couple dozen is the norm. So, they had no clue what this was, what purpose it served, what the repeats were doing, what the unique spacers were for. So they came up with a name, and essentially that was it. They named them, they named these clusters as clustered, regularly interspaced, short, palindromic repeats. And together, so C-R-I-S-P-R, CRISPR. So CRISPR arrays. And this would be one CRISPR array. The repeats are arrayed. So that's where that array comes from. So this is kind of a funny name, right? Clustered, because the repeats are clustered together in a certain location of the genome. Regularly interspaced, because there are these spacers of the same size in between the repeats. Short, palindromic, so inside these repeats there are often palindromic sequences. So little repeats that are the same forward, little sequences that are the same, read the same forward and backwards. So that's where that short palindromic comes from. That's what those little arrows are. I'm just indicating possible palindromes in these repeats. And the repeats, yeah, okay, so that's where the repeats come from. They're repeated sequences. So for, let's say, a decade, at least a decade, maybe 2 decades, they were just there in the genome. No scientists thought they might be important, but couldn't figure out what they were for. So it turns out that, they're really important. So let me diagram just a shorter array here, because I don't want to spend the whole video just making these very similar diagrams. This gives you some time to think, right, and take your own notes. Okay, so I'm going to add a few more things here, because now we know what these things are doing. So again, the dark colored sequences are the repeats. And you know, I want to put one more unique sequence in here. And this little line right here, it's double-stranded DNA. It's just another part of the genome. Now we're going to call this right here the leader sequence. We're going to call this right here the terminator sequence. And here are the repeats. And here are the unique sequences. So we now know that these unique sequences here, these are viral sequences. So they're sequences of viral origin. And so what are sequences of viral origin doing in a bacterial chromosome? You know, short sequences, about 20 base pairs long. What are they doing in the viral genome? So it turns out, let's say this is the bacterial cell wall and cell membrane. Just this whole thing right here represents both of those structures. And we have a bacterial virus, which is also called bacteriophage. That's the DNA in there. So the virus injects the DNA, and the virus's goal is to, you know, have this DNA transcribed into RNA, and the RNA transcribed into viral proteins to, you know, initiate or drive the viral infection to make more viral particles. Well, the bacteria, you know, has restriction enzymes. Bacterium, I should say, has restriction enzymes that, you know, look for certain sequences in viral DNA and try to chop up the viral DNA. But they also have other enzymes that will pick up bits of the viral DNA. You know, I'm not sure if it actually cuts the viral DNA itself or it picks up viral fragments on its own. I should look that up. But let's say this is a bacterial enzyme, and I'm going to give these guys a name in a second. Let's say it's a cluster of enzymes. So it's a complex of bacterial proteins. And what they'll do is they'll pick up a piece of the virus and they will drop it right in here. And if you're interested in the details of this mechanism, like at the DNA level, what the sequences look like, we cover that in BSc 350. Molecular biology. But we're going to just go over the general process here in a very conceptual way. So they pick up a piece of the virus. There's a fragment of the virus. You're going to have to trim it to the right size, right? About 20 base pairs. And they're going to drop it in here. And we're going to add another repeat. So this is called CRISPR adaptation. So what the bacterium is doing is it's building up the CRISPR array with pieces of viral DNA. So the longer the CRISPR array, the more pieces of viral DNA are contained in the CRISPR array. Now, CRISPR adaptation usually works by adding the newest sequences to the very beginning of the CRISPR array. That's where this L right here, this stands for leader. This marks the beginning of the CRISPR array, and the new sequences are added at the beginning. And so if we were to look all the way down towards the end of the CRISPR array, we have this T, this is the terminator of the CRISPR array. We could predict that this last sequence should come from an infection that happened a long time ago, possibly in an ancestor of the current bacterium. So in this way, the bacterium has a memory of all the viruses that, you know, infected it in the past, or most of them, assuming that, you know, a bit of that, each virus that infected it was integrated into the array. Also, it has a memory of the viruses that infected its ancestors. Okay, so what good is memory, you know, a sort of memory of the viral DNA that, you know, infected the bacterium in the past, or the bacterium's ancestors in the past? What good is that? What good is that? Okay, we're going to cover that in a second. So just let me point out, so past infections would be closer to this end. Recent infections, the viral sequences from recent infections, would be closer to the leader end. Okay, so what good is this memory? So it turns out that CRISPR array is transcribed. So let me put the CRISPR array in here again. CRISPR array. We'll just do a short CRISPR array. 1, 2, 3, 4, 5, 6, 7, 8, maybe 8 segments. I think I want to do that. We're going to have the leader sequence here, terminator here. Now let's start with the repeat. Okay, so here's our CRISPR array. And so it's going to be transcribed by what? So RNA polymerase, right? RNA polymerase, and that's going to give us, here's the transcription start site, here's the termination region, or transcriptional cleavage site. That's going to give us the, what we're going to call the precursor CR-RNA, for CRISPR RNA. So this is our RNA sequence right here. Oop, I didn't mean to do that. So here's the leader, here's a repeat. Now it doesn't matter what if we have a unique sequence coming right after the leader, or a spacer coming, or a repeat coming right after the leader. As long as we're alternating, we have the general idea of what these arrays look like. Okay, so this is an RNA, so a single-stranded RNA molecule, right? So this is DNA in the bacterial chromosome. RNA polymerase transcribes the DNA. We get a single-stranded RNA molecule called the precursor RNA. This is not translated into a protein. This is not translated. This is a non-coding RNA. It doesn't code for any proteins. So no ribosomes are going to bind to it. Instead, these are going to get processed by other bacterial proteins, and what they're going to be processed into are these things called CRISPR RNAs, right? So we have the precursor CRISPR RNA, and then they're going to be processed into CRISPR RNAs by bacterial proteins. Okay, so what do those CRISPR RNAs look like? Well, they essentially have the viral sequence, which is an RNA, a single-stranded RNA that is from the spacer sequences, the unique spacers, that matches the viral sequence that was put in there. And then they have the repeat sequence. So this part right here, remember those palindromic regions? Well, they tend to help this part of the RNA molecule form intramolecular hydrogen bonds and form some sort of hairpin secondary structure. So we've got the spacer sequence here and the repeat region here. Now these are RNA molecules, and the repeats are forming a secondary structure which is going to be recognized by bacterial proteins, and bacterial proteins are going to bind those things. And those bacterial proteins are going to have nuclease activity, so chopping DNA. So bacterial protein bacterial protein. I could call it an enzyme because it has a nuclease activity. Okay, so what are these going to do? Now, these are going to just search the cell for DNA sequences that match the CRISPR RNA, right? Now, what type of DNA sequences are going to match the CRISPR RNA that are going to have complementarity to the CRISPR RNA? Viral DNA, right? Because remember, during the CRISPR adaptation step, parts of the virus were taken and they were put into the CRISPR array. So now, when the CRISPR array is transcribed into the precursor CRISPR RNA, and when the precursor CRISPR RNA is processed into individual CRISPR RNAs, those CRISPR RNAs are picked up by bacterial proteins and used to scour the cell for viral DNA sequences. And if it should find a viral DNA sequence, the nuclease will chop that virus right where the CRISPR RNA is complementary to the virus, or at least one of the strands of the viral DNA. It'll cleave the viral DNA, and theoretically the viral DNA will not be able to make viral RNA. Viral RNA will not be able to make proteins, and the virus will not be able to infect that bacterium. So the CRISPR array is a vital component of defense against viruses. So viral defense or virus defense. Now, we talked about CRISPR adaptation was the process of adding new spacer sequences to an array, the spacers being derived from the viral DNA. CRISPR interference is the process by which bacterial proteins use the CRISPR RNAs to target viruses for cleavage. Okay, so now these proteins, whether they're involved in CRISPR adaptation or precursor CRISPR RNA processing or binding the CRISPR RNAs and looking for viral DNA to cleave, these proteins from the bacteria are called Cas proteins, for CRISPR-associated proteins. So the adaptation process, the interference process, those processes are mediated by Cas proteins, CRISPR-associated proteins. So it looks like bacteria have been, I should say, adapting the CRISPR process throughout evolution, or this sort of viral defense process through evolution. So we now know of 6 different types of CRISPR systems. The CRISPR system including adaptation and interference. So 6 different types, 6 different classifications we have for the slightly different types of CRISPR systems. We are going to talk about the type 2 system because this is the system that is used by geneticists around the world to engineer genomes, and it's a really useful system, and it's even led to the first genetically engineered humans, which we're going to talk about in, I think, a later video. Okay, so I will see you in the next video where we talk about the type 2 CRISPR system.